Nicole Leogas, 5/27/20. Today we will be talking about the determination of sex, genes versus hormones. So first we're going to start with some baseline terms. Gonads are the organs that produce gametes. Gametes are the reproductive cells that unite to form a new individual. Testes are the male gonads that produce sperm. Ovaries are the female gonads which produce eggs or ova. Germ cells are embryonic gonadal cells that produce gametes. Your internal genitalia consists of accessory glands and ducts that connect the gonads with the outside environment, and external genitalia, which includes all external reproductive structures. Sex chromosomes. Sex chromosomes are the main determinant of an individual's sex, and that's the sex chromosome that they inherit. Genetically, females are XX and males are XY. The Y chromosome is essential for development of the male reproductive organs. So sometimes the chromosomes don't always get distributed equally, so if they're abnormally distributed at fertilization, there's a few scenarios of what can happen. So if the zygote has a multiple X chromosomes as an XXY, the zygote will still become a male because it has the Y chromosome. If the zygote only inherits a Y chromosome, which would be YO, the zygote will die because it's missing important genes that are provided by the X chromosome. If the zygote inherits only an X chromosome and is XO, the zygote will develop into a female, but in order to survive, the female needs two X chromosomes to be able to have normal reproductive function. Differentiation. So before differentiation, embryonic tissues are labeled bipotential because they cannot be identified as male or female. The bipotential gonad has an outer cortex and an inner medulla. The medulla will later develop into testes, or in the absence of a developmental signal, it will differentiate into ovarian tissue. So that right there kind of tells you where you start to decide, or the cells start to decide if they will be male or female. Bipotential internal genitalia consists of accessory ducts, the Wolffian ducts and the Mullerian ducts, which later develop among male or female lines. Bipotential external genitalia consists of the following: the genital tubercle, urethral folds, urethral groove, and the labial scrotal swellings. The SRY gene. So this is an important gene in the determination of sex, depends on the region of the Y chromosome. In the presence of this gene, the gonads will develop into testes, and in the absence of this gene, the gonads will develop into ovaries. But it will only develop into female if there's other female-specific genes present, which are normally located on the other X chromosome. So for males specifically, their embryonic development, it's all about the SRY gene. The SRY gene produces a protein that binds to DNA and activates additional genes. Once the testes differentiate, they secrete hormones. Those are testicular Sertoli cells, which secrete a glycoprotein anti-Mullerian hormone. Testicular interstitial Leydig cells secrete androgens, testosterone and dihydrotestosterone, the dominant steroids that are present in males. If you look to the right over here, you can see how it splits off in the male development. So the sex-determining region of Y chromosome is the SRY gene. It produces testes-determining SRY protein, which initiates production of multiple proteins that cause the gonad medulla to differentiate into testes, which has interstitial cells and Sertoli cells, and those interstitial cells secrete testosterone, and that controls development of Wolffian duct into accessory structures and development of male external genitalia. The Sertoli cells secrete anti-Mullerian hormone, which causes the regression of the Mullerian duct. Lastly, we have the female embryonic development. So females, their embryos have no SRY gene. Instead, the cortex of the bipotential gonad develops into ovarian tissue. In the absence of testicular AMH, the Müllerian ducts develop into upper portion of the vagina, uterus, and fallopian tubes. Without testosterone, the Wolffian ducts degenerate. And without DHT, the external genitalia take on female characteristics. So overall, there's a lot of stuff that goes into deciding whether the embryo becomes male or female, and it has to do with hormones and the regulation of where those hormones are secreted.